Boon and Bane of ROS, how redox balance shapes cancer cell fate. Can targeting the delicate balance of reactive oxygen species, ROS, in cancer cells unlock new strategies for therapy, or does it risk fueling tumor survival and progression?
Redox balance, particularly the regulation of reactive oxygen species, ROS, plays a critical role in cancer cells. ROS are highly reactive molecules containing oxygen, such as superoxide, O, hydrogen peroxide, HO, and hydroxyl radicals, OH. In normal cells, ROS are produced as byproducts of cellular metabolism, primarily in the mitochondria, and are tightly regulated to maintain cellular homeostasis. However, in cancer cells, the redox balance is often disrupted, leading to elevated ROS levels, which can both promote and inhibit tumor progression depending on the context. Key aspects of redox balance in cancer cells. Elevated ROS in cancer cells. Cancer cells often exhibit increased ROS production due to oncogenic signaling, metabolic alterations, and mitochondrial dysfunction. Elevated ROS can drive tumorigenesis by promoting DNA damage, activating pro-survival signaling pathways, e.g., NFB, MAPK, and PI3K-AKT, and inducing mutations that favor cancer progression. ROS as a double-edged sword. Pro-tumor effects. Moderate levels of ROS can promote cancer cell proliferation, survival, and adaptation to stress by activating signaling pathways that support tumor growth. Anti-tumor effects. Excessive ROS can cause oxidative stress, leading to cell death, apoptosis, necrosis, or ferroptosis. This is why some cancer therapies aim to further increase ROS to toxic levels. Adaptive mechanisms in cancer cells. Cancer cells develop adaptive mechanisms to cope with elevated ROS, such as upregulating antioxidant systems, e.g., glutathione, thioredoxin, and superoxide dismutase, to maintain redox balance. The transcription factor NRF2, nuclear factor erythroid 2-related factor 2, is often activated in cancer cells to enhance the expression of antioxidant genes. Therapeutic implications. ROS-inducing therapies. Some treatments, such as chemotherapy and radiation therapy, rely on increasing ROS levels to induce cancer cell death. Antioxidant therapies. In certain contexts, antioxidants may be used to suppress ROS-driven tumorigenesis, but this approach is controversial because it may also protect cancer cells from ROS-induced damage. Metabolic reprogramming and ROS. Cancer cells often reprogram their metabolism, e.g., Warburg effect, to support rapid growth and survival, which can alter ROS production and detoxification. Metabolic pathways such as glycolysis, glutaminolysis, and fatty acid oxidation are closely linked to ROS regulation in cancer cells. ROS and tumor microenvironment. ROS can influence the tumor microenvironment by affecting immune cell function, angiogenesis, and extracellular matrix remodeling. Extracellular matrix, ECM, remodeling refers to the dynamic process by which the ECM, a network of proteins, glycoproteins, and polysaccharides surrounding cells, is continuously broken down, modified, and reassembled. This process is crucial for tissue structure, cell signaling, and functions like wound healing, development, and immune responses. In diseases like cancer, ECM remodeling is often dysregulated, enabling tumor invasion, metastasis, and angiogenesis by altering the physical and biochemical properties of the tissue microenvironment. Key enzymes involved include matrix metalloproteinases, MMPs, and tissue inhibitors of metalloproteinases, TIMPs. Immune cells in the tumor microenvironment, such as tumor-associated macrophages, TAMs, also contribute to ROS production and redox signaling. Challenges in targeting ROS in cancer therapy. Context-dependent effects. The role of ROS in cancer is highly context-dependent, making it challenging to develop universal therapies. Resistance mechanisms. Cancer cells can develop resistance to ROS-inducing therapies by enhancing their antioxidant defenses. Balancing act. Therapies must carefully balance increasing ROS to toxic levels without harming normal cells. In summary, redox balance and ROS regulation are central to cancer biology, influencing tumor initiation, progression, and response to therapy. Understanding the complex role of ROS in cancer cells is essential for developing targeted therapies that exploit redox vulnerabilities in tumors.